Okay, thank you.
Okay. Thank you.
Thank you so much. Now, Claudia Favara will present to us microplastics exposure in dialysis patients, preliminary results of a case control study. Claudia Favara is a PhD student of the University of Catania.
Thanks to the chair for introducing me. Today I'm going to talk about microplastics exposure in dialysis patients, preliminary result of a case control study. The birth of the plastics has brought great benefit to humanity and enabled some of the most significant advances of modern civilization in fields such as medicine, electronics, aerospace, construction, food packaging, and sport. In addition to benefits listed, we know that plastics is also main cause of significant damage to human health, to economy, and to environment. Microplastics smaller than 10 micrometer are potentially able to penetrate blood and tissue, and the human deal this particle through ingestion, inhalation, and dermal or skin contact. Exposure to microplastics is linked to various forms of inflammation, of immunological response, alteration of endocrine system, and alteration of lipid and energy metabolism, and other disorders as demonstrated in studies carried out on laboratory animals. Microplastics is a source of exposure to substances used as plastic additive, for example, bisphenol or phthalates, which have effect such as endocrine disruptors. Chronic kidney disease is a progressive reduction to the ability of the kidney to filter metabolic waste from the blood. Some patients are subjected to dialysis treatment. Disease is a technique to reproduce some kidney function to clear the blood. The blood passes through a capillary dialyzer where the blood is separated by a concurrent flow of dialysate using a thin membrane. The material composition of dialysis membrane includes basic cellulose or synthetic polymer. Our study, we investigated to microplastics exposure of dialysis patients compared to control. Both case 40 and control 17 were recruited at the University Hospital of Catania, and the blood sample from cases and control were analyzed using a method patent by the University of Catania for the extraction, identification, and quantification of microplastics smaller than 10 micrometer, using a scanning electron microscope combined with microanalysis X-ray. On this slide, I highlight the main division cases and control into males and females. In total, we analyzed 8 females and 6 males on dialysis groups, or with an average age of 18 years. While for the control group, we analyzed 7 females and 10 males, all with an average age of 65 years. The pool of patients under examination was given a questionnaire to fill out, which requested some simple information. From the answer given, we can see that the majority reside in the city of Catania, and they were all also asked about the marital status and the level of education. For control, 18% males and 71.4% females are married. For cases group, 100% males and 62.5% females are married. Regarding the level of education, 50% males had a middle school diploma, and 42.9% females had high school diploma. While for cases group, 50% males and 62.5% females had a primary school diploma, therefore a lower level of education. This study showed that the majority of women undergoing dialysis and control have never smoked, whereas the men are mostly ex-smokers. All in the control are the current smoker. For those with kidney failure and the smoker, the damage is more serious because, in fact, the smoking is an accelerator of this disease, which can lead to dialysis. On average, both case and control patients, dialysis patients and control, consumed around 2 liters of water a day. 100% of cases group consume mineral water in plastic bottle, but in the control, the case group, in the control group, consume mineral water 100% of males, females, and 18% of males, but 10% males consume tap water or mineral glass water. Regarding eating habits, we see that the majority recruited case and control do not eat the food in the microwave using a plastic bottle, plastic container or plates, but still consume canned food or bagged food. In terms of concentration of microplastics found, female present high value than male in both groups. In the dialysis group, however, the concentration is high than microplastic found control for both sexes. The average diameter of the particle was found smaller in women and than in men in control group. In case group, the trend, we have an inverse trend. The project highlights preliminary data on the presence of microplastics in blood sample of patients with chronic renal failure and undergoing dialysis, and comparing them with healthy subjects. Through administration of questionnaire, we want to find a possible correlation between lifestyle, eating, and smoking habits of patients and microplastic concentrations. Cases in control had comorbidity and were therefore treated with various pharmacological therapy, which in addition to dialysis could be a source of exposure to microplastics. The response of contamination of ocean, river, lake, air, soil is inexorably linked to contamination of food and human, with negative health consequences. These topics still require a numerous study to fortify this preliminary data. Thank you for attention.
Thank you, Claudia.
Any questions?
Any questions for Claudia? Yes, one. Yeah.
Good morning. Thank you for your presentation. I wonder if you thought about if these patients that are doing dialysis, your results apply to every person, or do you think they have special conditions that make the results different from the remaining population? That's my question.
The result in this study is preliminary, and I research in other population, but the pool of the patients is very small, but statistical data.
I mean, if there are other conditions or special practices that lead to these results in dialysis patients, or if this is something that you see through dialysis results, but it applies to everyone. That's my question. I'm not sure if I'm clear. Do you understand the question? No. No. Sorry. So you are analyzing patients with a disease, right?
We have chosen different disease. This is the results of the study on dialysis patients, but we have studied patients with chronic intestinal disease, with colorectal cancer, and with major depression, and the study are ongoing, and the results at now are few because not all the sample are analyzed, but in the sample that we analyze, very often we try in blood and urines and tissue some plastic particles. So the nanoparticles go inside our body, and we are trying. The next step is to search how much these nanoplastics are correlated with the disease. And if there is a link between microplastic exposition and disease and progression of disease in the human. And for this, we are studying not only the presence of microplastics and the lifestyle with questionnaires, but we are studying in collaboration with Professor Izzotti the microRNA pattern of the tissue and for in normal condition in overexposition of plastics to create a possible pattern expression of microRNA linked with microplastics and eventually linked to the microplastics related to the pathologies.
Okay, thank you.
Any other questions? I have a question. Have you looked into perhaps working with children to see if maybe there could be any kind of correlation between the adults and the children that have dialysis?
Voglio sapere se c'è correlazione. Sì, rispondi tu, fra i bambini e i grandi. Ma in dialisi stiamo vedendo...
In dialysis solo, cioè,
Only
adult human and men.
We studied the only adults in this moment.
Okay, but have you considered in the future?
Yes.
Yeah.
Thank you very much for this very nice presentation and for the effort that has been and will be also performed by Margherita and Gea, identifying source and dissemination and most of all the effects. So the presence of microplastic in blood, but would it be possible also to make some biopsies? And probably my question for Margherita and Gea, and try to make correlation between specific diet for specific peoples with the number of microplastic that will reach the body, and then probably the disease. I know that we have performed some microplastic analysis in testis biopsies with Sergio Minucci because we have the support of the Policlinico, but also you supported by the Policlinico of Catania. Will it be possible to draw this kind of relationship between diet, typical diet? I am referring to our very nice project, Fontam, that we have built regarding the exposure and the effects. So probably we can proceed with this. What do you think, Margherita and Gea?
Yes, we are studying the relationship within diets and plastic presence and disease in these patients. We have the lifestyle, the diet habits with questionnaires, so we are studying this link, but the study is poor at now because we have a few patients. And we are taken more patients, and we are in progress with the nanoplastic lecture with Sam, and we want to have almost 30 patients and 30 control, or more if possible, for to do a more good analysis, because few patients make not significant the analysis, but is still in progress in the initial data, and we are studying the tissue. We are biopsies by patients with chronic intestinal disease, bowel disease, and tissue from colorectal cancer that our colleagues of surgery in Policlinico have picked up for us, and we pay attention on this operation to don't contaminate the sample. We have the blank sample, and for to have a good treatment of the sample and not contaminate the sample, and to have a comparison to to make a comparison with the sample, the blank, and also standard sample, because we have made the standard with standard plastics, and they have set and standardized the methods on the standard plastics, and then we have to recognize and to characterize the nanoplastic that we try in the urine, in the blood, in the tissue on human. It's a new scenario, a new research. There are no data on literature, so we are going step by step to search if eventually there is a link, and if there is a link to prove that this link and the pathology, the progression of the pathology have a relation with the nanoplastics.
Thank you very much. I would have just a second question, just to bridge the gap between the science and also the society. Will it be possible, according to your experience, because I know that you have a very well-equipped lab dealing with the identification, because people are filtering samples, but Jaya is not filtering samples. She is considering all the content, and this is a very important issue. Will it be possible to draw a certain kind of short list of diet that will be the most contaminating for people considering plastic? I mean, we know that consuming certain diet, Mediterranean diet, would be beneficial in somehow, but other diets will be promoting fat, etc. Will it be possible to draw a certain kind of short list of products that could be the most endangered and carrier of microplastic to humans?
Yes.
I reply with several examples. Because as you can see in the slide, we have a difference on concentration between women and male. And this can be for different habit styles, including foods, cosmetic uses. And another problem that can be releved by a reviewer of methodological epidemiological study design is how you select the control. Because we must to have only a robust design for epidemiological study. So we used as control the same population collected for diagnosis. The diagnosis positive for cancer, for major depression, for other pathology was selected as case. And the negative diagnosis was control. So it's possible that the entire population have the similar problem, the similar effects, but not is correlated of plastics. So we can demonstrate, I hope, at the end of the epidemiological study, to define a better diet habit, a better choice of things on plastics that we can use journally. But now is in this moment we cannot to say that this yes, this no, but at the end the study is possible because we have only all information about lifestyle, work they do, or type of water that they use. Because I study in this moment, I'm studying in this moment the difference between drinking water by tap water and mineral water. And we have a large difference in concentration, but in dimension. When we read the literature about blood tissue concentration microplastics, is normally to found 25, 50 particles between 10 or 20 micrometers. But is the the high level of the icebergs. Using our methods, we are able to detect for first time the micro and nanoplastics that are most abundant in the blood and in tissue.
Under 10 micrometers and under 1 micrometers too. And the other authors don't see this particle because they filter. We not filtered, so we recognize all the nanoparticles and all the microparticles, all the particles present in our samples. So we see much more particles in our sample. But all is the nanoscale at this moment is few studied. There is few study and few data, and so we have to reinforce this data. And in addition, we have to make on this patient at the end of the study a multivariate analysis, not only consider the lifestyle such as smoking, diets, and water consume, and the jobs and so on, cosmetic use, but also considering the zone, the areas where they living, because the exposition is very complex. The nanoplastic exposition is very complex by air, by respiratory tracts. So the nanoparticles go inside between the lungs, but also directly with the air inside the brain, because the olfactory cells with determination are able to transport directly nanoparticles inside the brain, not also with the lung absorption. And then we have another way, that is the dermal absorption. There is for the smaller nanoparticles, the dermal absorption is also to consider. So it is very difficult, and we hope in the near future to have more data and the possibility to have a good multivariate analysis considering all the variables such as is possible to do this.
Okay? No other? Okay. Thank you so much.
Thank you, Claudia, and now Hannah.